Record.
Thank you. All right. Hello, everyone, and thank you for coming. Today, my colleague Peter and I have for you the biosynthesis, synthesis, and anti-cancer activity of bis-benzyl isoquinoline alkaloids, which I'll be referring now to as Bbi alkaloids. So we chose this project because we work in an organic synthesis group, and for the past year or so, our group has been focused on synthesizing these two compounds, tetrandrine and berberine, which have been used extensively for decades in traditional Chinese medicine to treat a variety of illnesses. Today, we wanted to discuss our research on the review that we've been conducting over the past year or so on the structure, biosynthesis, and classification of these compounds, as well as some of the molecular mechanisms of their anti-cancer activity for both tetrandrine and berberine. And lastly, some structure-activity relationships for each of these compounds and specific proteins that they interact with. Bbi alkaloids are thought to consist of a head and a tail. The head is made up of the A and the B ring, and the tail is made up of the C ring and this alpha carbon here. These structures are dimeric in nature, and when we combine two of these monomers, we get a dimer of these subunits, and they're linked via a di-aryl ether linkage. Typically, the largest synthetic challenge with these compounds has been the construction of this di-aryl ether linkage. Currently, the best known method is the Ullmann coupling using copper bromide, but even this method suffers from low yield, which is especially detrimental in the later stages of organic synthesis. However, enzymatic reactions are able to traverse this barrier to produce these Bbi alkaloids in higher yield and facilitate this difficult bond formation. The biosynthesis of these compounds begins with the amino acid tyrosine, which is hydroxylated by tyrosine hydroxylase. Notice the new bond formed in red. Hydroxytyrosine is then decarboxylated by tyrosine decarboxylase to give hydroxytyramine. Hydroxytyramine is then combined with another organic compound via an enzymatic reaction conducted by norcochlearine synthase to produce S-norcochlearine. Notice the new carbon-carbon and carbon-nitrogen bonds formed in this step. The next step is the methylation of the 6-oxygen as well as the nitrogen in our compound by 6-O-methyltransferase and cochlearine N-methyltransferase to produce S-cochlearine. The final and most important step of the synthesis of Bbi alkaloids is conducted by berberine synthase, or cytochrome P80A1. This key reaction is able to traverse the synthetic barriers faced in the di-aryl ether linkage formation to produce berberine. One of the hallmarks of cancer is the ability of these malignant cells to grow and spread uncontrollably. Tetrandrine's value lies in its ability to arrest the cell cycle. In the cell cycle, there are two important checkpoints, the G2M checkpoint and the G1S checkpoint. Today, we'll be talking mainly about the G1S checkpoint. This checkpoint is traversed with the help of a complex of proteins called CDK4/6, as well as cyclin D. This complex is inhibited by the action of P21 and P27. Note that the crossbar indicates inhibition. These inhibitory proteins are activated by P53. Note that here, the arrows indicate activation. Tetrandrine intervenes far upstream of this pathway by activating P53, which will ultimately lead to decreased expression of cyclin D, which seizes the cell at the G1S checkpoint. This was demonstrated in 3 cancer cell lines, but today we'll be talking about the HepG2 cell line, which is cancer of the liver. In a concentration-dependent manner, tetrandrine was able to reduce the amount of cells in the S phase. At 0 micromolar tetrandrine, we see that 49% of the cells are in the S phase. When we increase the dosage to 3.2 micromolar, we see that only 17% of the cells are in the S phase, which is a significant drop-off. To understand how this works, we have to consider the protein cyclin D. Cyclin D is expressed under the control of a biological pathway known as the beta-catenin pathway. When cells are not dividing, beta-catenin is bound by an inhibitor. When cells are dividing, the IGF receptor inhibits the inhibitor, allowing beta-catenin to increase expression of cyclin D. Tetrandrine is able to inhibit the IGF receptor and ultimately lead to the inhibition of beta-catenin and decreased expression of cyclin D. Note that exogenous IGF binding protein is able to restore beta-catenin to near baseline levels, which confirms the proposal put forth in the International Journal of Oncology. This occurs in a concentration-dependent manner. One of the difficulties in treating cancer lies in the ability of the cancer to evade chemotherapies. One key mechanism by which this occurs is by the overexpression of a drug export protein known as P-glycoprotein. In this case, we've illustrated a subfamily of this protein known as ABCG2. We learned previously that tetrandrine is able to activate the inhibition of beta-catenin. Beta-catenin is normally responsible for activating expression of P-glycoprotein. So you'd imagine that tetrandrine is able to reduce P-glycoprotein action and expression and reduce the efflux of paclitaxel, a key anti-cancer compound, from the cell. In a single dose strategy, tetrandrine alone has a half maximal inhibitory concentration of about 8.5 micromolar. However, when combined with paclitaxel, a known anti-cancer drug, this IC50 value drops to 1.5 micromolar, indicating an increased potency by about 6 times, which is highly significant. Several proliferative proteins that are involved in the growth and division of cells were also seen to decrease in the combination treatment strategy, as well as an increase in the inhibitory protein P21. This occurs again in a concentration-dependent manner and sees increased effects when in combination therapy as opposed to solo therapy. The key to the interaction of tetrandrine with P-glycoprotein lies in its structure-activity relationship. TET is able to bind in a hydrophobic binding pocket deep within the P-glycoprotein. This has a docking score of negative 9.7 kcal per mole, which is significant because a score beneath 9 kcal per mole indicates a strong binding, further supporting the hypothesis that tetrandrine is able to inhibit P-glycoprotein expression. This interaction is stabilized by pi-pi stacking of aryl rings, as well as cation-pi interactions between the amine in tetrandrine, as well as aryl residues inside the protein.
As Yaseen has mentioned, berberine is also an interesting anti-cancer agent. Today, we'll be touching up briefly on its biological implications, structure-activity relationship, specifically its binding to certain proliferative proteins. We can learn a lot about drug delivery and metastasis from the use of berberine. So what these researchers here did is that they used a scratch assay to scratch the membrane of the cell and see how long it takes for that cell membrane to heal. And it turns out that in the presence of berberine, these cells heal a lot slower, showing its anti-migratory effect. And that effect was exaggerated even more by the delivery of berberine in lipid nanoparticles, which increased the delivery at the target tumor site. And that led to more cells dying or being arrested during cell division, which further shows the anti-migratory effect of berberine, as well as its efficiency in arresting the cell during the cell cycle, and how that effect is exaggerated when the drug delivery is improved, which is one of our aims, is to we hope to derivatize these molecules after synthesizing them so that when we deliver them, we can increase the efficiency of delivery. And moving on from here, I'll be talking a little bit about structure-activity relationships. So berberine has been shown to decrease the amount of phosphorylated STAT in the cell. And what phosphorylated STAT usually or normally does without berberine is that it enters the nucleus and leads to the expression of proliferative and survival proteins. But when you inhibit that phosphorylation using berberine, then you don't synthesize or don't express more proliferative and survival genes, and therefore the cell gets arrested. And that's shown by this Western blot on the right. As you can see, BM for berberine, as you add berberine, you decrease the amount of phosphorylated stat, which leads to less expression of cell survival and cell proliferation proteins. And moving from there, I'll show you guys the actual docking studies that these researchers performed, showing the hydrophilic interactions between the stat protein and berberine. And these hydrophilic interactions block or decrease the amount of stat that can be phosphorylated, which is shown also by these figures on the right, that as you increase the concentration of berberine, you get less phosphorylated stat. And when you have less phosphorylated stat, you have less survival and proliferation. And that's also shown by this figure all the way on the right, that as you increase the concentration of berberine given by 5, 10, 20, and 40, you decrease the amount of phosphorylated stat. And this is what we had for you guys today. And with that, we'll take any questions.
Thank you all.
Thank you. So I encourage anyone from the audience who'd like to ask a question to unmute yourself.
I have a question in regards to to Yaseen, in regards to the whole molecular pathway in the inhibition of the molecular pathway of the inhibition process. Can you go back to that slide of TXT? Yeah, the slide here of TET. So with the binding of TAT, does it cause disinhibition from the IGFR to the inhibitor for beta catenin? Or
sorry, go ahead.
Yeah, is it disinhibition or stimulation?
So this is a sort of complex pathway to consider because we have multiple inhibitory events and we're essentially inhibiting an inhibitor. But I think what sort of helps to clear this up is to consider the fact that in a non-dividing state, beta-catenin is bound by this inhibitor.
Mm-hmm.
And when the cell is dividing, this IGF receptor inhibits the inhibitor. So that effectively takes the inhibitor out of play and allows beta-catenin to express D1 or cyclin D1. And tetrandrine is basically able to inhibit the inhibitor of the inhibitor. So it's quite a mouthful and it's quite difficult to think about, but when you take it piece by piece, I think it makes a little bit more sense.
All right, thank you.
No problem, thank you.
I have a question.
Go ahead.
Yeah, hi, thanks Yaseen, and it was a great presentation. I have a question. What are some of the challenges associated with implementing this treatment to patients who have cancer and have developed cancer resistance to chemotherapy?
So that actually is one of the strengths of this combination therapy. So this paper basically outlined the effect of this combination therapy on chemotherapy resistant cells specifically. So the cell line in this study was an ovarian cancer cell line that was exposed to a gradient treatment of paclitaxel or PTX. So what they do is they slowly increase the concentration of PTX over a long amount of time to make these cells resistant to the drug. So this is also seen in motifs such as antibacterial resistance or antibiotic resistance. So in the clinics, oftentimes doctors might overprescribe antibiotics and these bacteria develop into resistant strains. And that's basically the same principle at play here, where the cells, the ovarian cancer cells, develop a resistance to paclitaxel. And tetrandrine is basically able to resensitize these cells to the original treatment. So note that in the combination treatment, we see a significant drop-off in the amount of protein expression. And these are proliferative proteins. And this final protein, P21, is an inhibitory protein, which prevents cell growth. So this sort of paints a very strong picture that this drug has promising anti-cancer activity, which is why it's been used so long in Chinese medicine and why it's such an active area of study in chemistry and biology today.
If I may add to that, Yaseen, yeah, another reason why we're really interested in these molecules is that the whole class or most of the class of these molecules from what we've been researching seems to have that same effect. Like berberine seems to have a similar effect, so does neferine. And that's why we're interested in these molecules, because even neferine has shown to be more selective with healthy lung cells versus cancerous lung cells, and which ones they put in cell cycle arrest. So the toxicity or the side effects from these alkaloids seems to be less detrimental.
Hope that answers your question, Shaker. Thank you.
Yep, thank you guys, appreciate it.
All right, so there's time for maybe one more question. It can either come from the audience, or if the audience is feeling shy, I can ask it. All right, so was there any aspects of this research project that completely surprised you?
I think for me personally, the biggest surprise is sort of the commonalities that we saw between all of these compounds. So today we discussed only 2 of these compounds, but there are hundreds of Bbi alkaloids, and they all seem to share a lot of the same properties, which is why Peter mentioned that this is such an active area of study. We see the same sorts of bioactivity with varying degrees of strength in each of these compounds, but it is a very hot area of research right now for a lot of different fields. And this is kind of why Peter and I decided to embark on this interdisciplinary, multidisciplinary study to sort of compile all of the data available and put out a very comprehensive review so that there is sort of like a singular site where people can read up on these compounds and make decisions, make research decisions in their labs.
Yeah, for me, I mean, the biggest thing was how interdisciplinary this project is, because Yaseen and I enjoy cancer biology, we enjoy biology and chemistry, and we found and saw this project is kind of the perfect combination of all these things, and through that we did learn a lot more compiling these review papers and putting these things together. We learned a lot more that we weren't able to learn in some of these classes, and we strengthened some of the knowledge that we learned here at UCR from some professors about these certain topics.
Okay. Perfect. So I'm going to remind you now to stop recording.